to the computer. And now we're going to share our PowerPoint. Everybody see that? Good? No. Put those on the side. There we go. All right, so cell flow cytometry. We got a glimpse of this. Like I said, when we introduced it, we watched a video of To Save a Life. And they were talking about how, flow cytometry had been used to help diagnose a young man's cancer, the specific type. And once they found the specific type using cell flow cytometry, they were able to treat it based on that diagnosis. And then they were able to rerun the blood and to see that those changes had been eliminated. So the cells had been, there was a mutation in his cells where he had a translocation, a specific translocation of his genes, where there was part of one gene was in on a wrong chromosome and the other part was in another chromosome. And once they eliminated those cells, then they were able to kind of restart his cells back to normal. But they had to get rid of that translocation, all the cells that had that. So that's what the treatment was for. Cell flow cytometry has not always been in the lab. I've never worked with it in the labs, a clinical lab, but your text will tell you that it was introduced maybe in the early 80s. It's been around for a long time. In the 1960s, the technique was there. And one of the, when you hear cell flow cytometry, you think, oh, this is some kind of complicated method. And it is kind of complicated as to counting cells and to see the certain cells and to detect certain cells. But we do this with hematology. And I'm just kind of curious as to how much analyzer info you got on how the analyzer actually works with the differential. Any at all?
Not really. It was more of a, hey, these are the buttons you press and these are the controls you use.
Okay.
And that was
nothing about how it actually counts cells, how it lyses red cells away and then counts white cells and puts them in a scatter.
We talked about it, but I don't remember.
Scatter plot, maybe. Did y'all talk about a scatter plot? Did she show you a printout of the CBC analyzer and show you kind of a scatter plot to the side of the population of cells? Kind of see that, and you kind of wondered how they came up with that and how they knew that those cells were, like how did we count our monocytes and how do we count our lymphocytes? How does it know? How does it know that's a neutrophil? How does it know that's a lymph? How does it know it's a mono? How did it automatically do a differential? And some of that is based on this technology. So when we say it hadn't been in the lab and it's not part of the lab, it kind of is when we think about a hematology analyzer and how we do a differential. So we're going to talk about the instrument itself a little bit, how the data is acquired, and a computer is needed. Okay, so you're not going to be like, oh, I got the counts and now, so a computer will analyze. You can control that a little bit on how you box the scatter to analyze the amount of the cells. And then the last part is immunoassay automation, and that we just have a couple of definitions that you should be familiar with on that. So we're going to spend most of our time in cell flow cytometry. So cell flow cytometry, automated process, analyzes cells or beads, so that in the fluid suspension for their light scattering characteristic. So you use a laser, okay? You use fluorochromes. So usually a hematology analyzer has some kind of dye pack. If they showed you that, you have to change it out every so often. But what it does is it mixes with your blood, and it will dye the cells so that they can, when they go through the counter, they drop through one by one, that they can be counted, okay? So it allows for the particle size, shape, and antigen properties, and that's where we use it for flow cell. is that we can actually check antigens. You know, the CD story, the cluster differentiation story, kind of concludes here, because we talk about how we detect all the different CDs on the cell and what that means. And we introduced that last Friday with leukemias and lymphomas and the characteristic cells. How did we know those are the cells that are present? How do we know it's the CD54B cell? Well, we use detection. We look for that marker. We look for that distinct, you know, differentiation on the surface of the cell, and that allows us to make the, if you want to call it, the ID of the different leukemias. So it is rapid, it is accurate, and it finds cells in small numbers. So you think about a, you know, white blood cell count, you know, of what, 10,000 per cubic millimeter of blood, something like that. So we don't have to have millions of these cells. So the use of cell flow cytometry, it really came into the early 80s as a tool for HIV infection, identifying HIV infection. We see it with immunophenotyping. We see it with diagnosis of leukemia and lymphoma, and that's why we put it at the end here, because that's what we just got through talking about Friday. And functional assays for various conditions. There's other things you can use it for. But with HIV, one of the things that we're looking for is the CD4 count or CD8 count, the T helper and the C cytotoxic differentiation count on those. So we can actually monitor the T helper level based with cell flow cytometry, count the actual lymphocytes that are T helpers. And see what that number is doing. If it's an acute HIV infection where you're losing those numbers, or if it's chronic where you lose them over time, or before you develop AIDS. So that's definitely a useful tool with monitoring HIV infection. So here's the instrument itself. So you see that the cell suspension, and they talk about, you know, you just draw up a tube of blood and EDTA. All right, so that is cell suspension and liquid suspension. As it comes through, what you want is one cell at a time. So you can imagine the size of this sheath bringing the fluid down. You got a laser that's coming in here, and as the cell drops, it breaks the laser beam and it allows it to count. Okay, and then based on the scatter of that cell size, fluorochrome attached, all of that good stuff gets knocked around. If you see the dichromic mirrors into detection. So all the different ways light is reflected or deflected, then it's detected by, what is that? Looks like 4 sensors maybe. 5 sensors. You see a forward angle light scattering, you see a side scatter detection, a fluorescent detector, and 2 others that aren't even labeled up there, and sends a signal to the computer, and the computer tells you, blah, this is what we got, right? So I don't expect us to know, like, be able to build one of these or to tear it apart and understand everything about it physically, the physics part. That's not what we're wanting to do, but we're wanting you to kind of introduce what the component is, like what is the technique. So we have intrinsic and extrinsic parameters, optics and photo detectors. So a lot of fun stuff if you love light physics and how that works. So that is the actual setup. This is 195 in your text. So what do we do with the
question?
What's that?
I have a question.
Okay.
In the review quiz, there was a question, the very last question that I had on it the first time I took it, and I answered forward angle light scatter, and it was wrong. Do you want me to redo the question? Because I was confused as to why it was wrong.
You can. I don't know which question it would be. I don't have the you can go ahead and shoot, see if I got it here.
Okay. It was which light signal in flow cytometry could be used to provide information about the expression of a protein on the outer surface of a cell membrane.
Okay.
I put forward angle light scatter. It might be wrong, but I thought it was right.
All right. That must have been, don't see that question here. So it must be coming from the test bank.
Yeah.
We can look at forward right quick. Did you look it up?
Yeah.
Okay, so what page are we?
It's just the very first one, 194.
Okay. So forward angle and side scatter size, indicator of size, whereas side scatter is indicative of granularity. So these two are called the intrinsic. So you're looking at extrinsic light scattering would be the additional fluorescent to tell you the antibodies labeled or proteins labeled onto the cell. Was that a choice?
No, it's
Yeah, what were the choices? Because I don't think it's forward or side.
it was forward, right, red light emitted by phycoerythrin, or polarized light.
Oh, well, I know it's going to be here. No, it's probably going to be the red
Okay.
as your one, because that's going to be the light that detects the fluorescence. There's red, blue, and violet, I think, are the different colors that come out. That would be my guess. But that sounds like a question out of the test bank.
Yeah, it was on the review quiz. The next time I'll check it, if it pops up, I'll put that.
Oh, I'll look it up. I'll look it up and send it out.
Okay.
I'll look it up. I just don't have those. I put those straight into Blackboard Friday. I didn't even hardly.
The right
look.
answer. You don't have to look it up. That is the right answer. That's the one I have on my Quizlet.
Is the red?
Yeah, that's the one.
Okay, awesome.
Okay. So yeah, sorry that, I mean, the technique is that the scatter is size or granularity, kind of like what we do with the diff of a white blood cell count differential, but to actually detect a certain cell based on a protein or, using an antibody, looking for a CD protein out there, then yeah, it would be more.
Okay.
It'd be the fluorochrome
Sorry.
part. The reason you mix it with the fluorochrome. All right. Thank you, Mary, for your help. All right. So what was the M1 here? Y'all remember that? Didn't we have that Friday? M1 popping up, maybe in the electrophoresis of one of our leukemias, maybe. I thought M1 was familiar, but maybe not. I'm sorry, Discovery Card wants to sell me a credit card. See if I can mute that. Oops. Okay, sorry. I mean, when I see M1, I think that this is a description under it, doesn't say much about it. It just says a count and fluorescent intensity. And then all of a sudden it has this M1 peak. Does anybody remember that from lecture last Friday? chapter did we have last Friday? 18?
Yeah, it says that M stands for monoclonal.
Okay. Is that, did we have the M peak in our things? The M spike, I guess is what I'm referring to. The monoclonal.
I'm not finding it. I'm finding spikes and albumin, polyclonal. Well, we have monoclonal on figure 18.6 on 318.
Yes, that's what I'm referring to. Okay, I was just looking at the histogram here. This is more about what I'm thinking of your CBC, a dot plot. So as you see, there's areas of counts in certain areas. So if you're, the idea of a scatter plot showing you the area of neutrophils, so you would kind of like your analyzer would look at the dots up in this area, red blood cell count, lymphocyte count, monocyte count. So you can kind of see how a scatter plot, as it adjusts, that's how your analyzer actually goes about determining the amount of cells in the CBC. So I know it's some of the, I didn't know how much of that you got early in heme. You maybe should have watched a video on hematology analyzers and how they actually operate. So I don't know if you want to do that. I can't even remember the name, but I know Sysmex would have something here how it actually operates. So here's some more ideas. This would be like for our CD4 count. So, we're not going to get too deep into this, but quadrant analysis or dual parameter dot plots. So based on the scatter, based on the detection, puts this cell into certain areas. Okay, so the fluorochrome attached to a CD4 puts it in a certain area when it goes through the flow cytometry scatter. So based on the laser hitting it and the fluorochrome, adjusting the color, intensity where it gets scattered out, it gets detected, it hits. So I'm just showing you some examples here. I don't want to get too deep into the physics, but I do want to talk about how you would prep. So this is more, real world, whole blood, EDTA. That is the purple top, lavender top test tube. If you don't, the blood tube, if you don't know that by now, hopefully. Stored at room temp, 20 to 25, should be mixed well. And then you run it through. The peripheral blood, bone marrow samples with large number of red blood cells require erythrocyte removal. So you kind of do how do you remove erythrocytes? You lyse them. So you put it in a lysing agent, and your hematology analyzer does that. It lyses red blood cells. Once it does a count, it frees them out, lyses them. Some of those reagents that you have in that machine are lysing reagents. So they lyse the red blood cells before you do a white blood cell analysis. And you can actually do this manually. I think they were called OPETs. We used to do these just to show you. I don't know if Miss Annette does or Miss Bednar does this, where you can manually do a white blood cell count. Y'all do that in Heme 1. And you have to lyse the red blood cells to do it. Makes it a lot easier. I don't know if she did that or not. But they're called OPETs, and they're just a manual little solution in a little plastic vial. You put your blood in there, the red cells lyse out, and then you load your hemocytometer with that, and you have your white blood cells on the hemocytometer to count with no red cells in the way. So it allows you to free up and see those. So it's pretty good, pretty neat. If you had a tissue, if we're doing like, you should be collected, transport, tissue culture medium, room temp, should be disaggregated via mechanical dissociation or enzymatic digestion. Should have antibodies, usually monoclonal, added before processing, because if you destroy the tissue, then you don't get the detection of the antigens on the tissue. So what do we use it for? What is the clinical application? So this is like on 197. And this is where we're going to spend probably the rest of the time, and then we'll hit on accuracy and precision at the end. Identifies particular markers for diagnosing and monitoring leukemias and lymphomas. And you're talking about a list, right? We have surface markers on the leukocytes. This starts on page 198 and goes basically, well, the antigen and cell type to 199. So you can see that we can move through with the antigen, those CD markers. We start at 2, 3, 4, 5, 6, 7, 8. Then we pick up at 10, 11B, 13, 14, 15, 16, 19, 20, 21, 22, 23. So all those numbers that we've played with, especially last week, they're here, right? So this is how you would go about detecting if those cells were present, the particular B cell, T cell subsets, plasma cells. You know, we can test for those markers and detect those cells that have those markers based on this idea of just flow cytometry. So instead of having to pull the cells and put them in with a certain monoclonal antibody and hope it didn't cross-react, you can actually do it a little easier with the application of a single cell going through the cytometry one at a time and seeing what happens. Okay, so then it gives you your counts. So you can do counts for the leukemia lymphomas, typing, you know how we did the diagnosing on Friday. It would tell you, you know, hey, this leukemia has a certain cell. This lymphoma has a certain cell. And we go about finding those with this technology. We can do counts of the peripheral blood, CD4 T cell. This is for HIV. We've harped on that just because that's the most used aspect of this technology. We can do CD34 stem cell transplantation, right? That's that hematopoietic stem cell that doesn't know what it wants to be when it grows up. So when you transplant stem cells, and you see this from what? You see this with NBA players. I don't know if y'all know this or not, but when they talk about getting knee injections, they're injecting stem cells into their knees to try to create an idea of cartilage cells, trying to heal their joints so they can keep playing basketball. So we see that used often. We can determine DNA content of some of those leukemias that are ploidy status. You remember if I had too many duplications of the chromosomes, we can definitely see the DNA content of a cell, and it also helps diagnose inherited diseases. So we have a lot of use. So what I want to do is open, if you don't have your books, there's 2 pages of cells of diseases. So this is not in the PowerPoint. I don't think I have it in the, oops. Yeah, this is just a slide we're going to start with. If you look at page 200 and 201, remember just kind of reviewing what we did Friday. We have chronic leukocyte leukemia, CLL, and polymorphic, or prolymphocytic, let me get my eyes straight here, prolymphocytic leukemia. The antibodies are paired. You see CD5 with CD20, okay? You see CD19 with kappa and CD19 with lambda chains. CD45 with CD14. And you can see the interpretation when a cell is both CD5 positive and CD20 positive. This is characteristic of CLL and well-differentiated lymphocytic lymphoma, as well mantle cell lymphoma. CD19 positive with only light chains is expressed in low intensity. Occasionally light chains are not detected. CD45 fluorescence brightly expressed with CD14 negative. Let's look at ALL. Let's go to B cell. CD5 and CD20 again. You see CD19 with kappa. You see these different types of problems, right? You see these different detected antibody pairs. CD3 T cell receptor negative for DR positives. Let's see. We have myeloma. We have T cell acute lymphoblastic leukemia, ALL, again with T cell versus B cell. And which one's more common with ALL? Oh, is that a question? Which type of ALL is more common, B or T?
I think it's B.
I think it's B. A B, good think. Yes. All right. So we want to finish up with this immunoassay automation. So immunoassays have been kind of moved to the analyzers, which makes it, we've been doing it, on the tabletop. And what I really want to work with here is this idea of accuracy and precision. I want to make sure you have that down. So if somebody would help me, what is accuracy?
Isn't accuracy like how many times you can do it over and over and over again and get like the same result?
You're a little ahead of yourself. That's coming up with precision. Accuracy is how close is my result to the real result. Like when we do a test, how close is that sugar glucose? How close is that to the real glucose in the blood? If you're not accurate, and that's where we, everybody talks about what it takes to be a medical lab scientist. Accuracy and precision, we're obsessed with that. So when we see new tests come out and they go, hey, it detects at 70%, to us, that's not very accurate. That means 30% of the people are being missed or getting the wrong result. So we're obsessed with accuracy, which is what is the true result? How close are we to the true level? So we are very, very obsessed with that. Precision is, can we do it again and again and again? And that's what Allison was saying, was precision is, can we do it 5 times and get the same thing? So can we repeat it? So it doesn't do us any good to get a blood sugar of 100 right on the bat, right? That's what the patient's blood sugar is. And then turn around and do it again and we get 50. And then we do it again and get 150. And then we got to do it again and get 100. So like 2 out of 5 times we get it right. That's not very precise, and that's where precision. So we have those two aspects of clinical lab, and you probably, I know you've had it covered in principles. You probably just didn't know what it really meant when it came down to doing tests. Because if you didn't do any tests, then you didn't know. So we had some really good tests, right? And that's what's going to lead us into our next part, which is reviewing of the labs. So on the midterm exam, where those that were really good note takers and really looked at the syllabus, where did we cut off the lab exam? Do you remember? So I know we didn't do the immunoelectrophoresis yet, right? Did we do the ELISA on the first exam?
I know we had it on there because we had to define like what ELISA meant.
Okay, so I think we got through ELISA.
Yeah, the only 2 things that were on the midterm were the dilutions and the ELISA, because there was only 4 questions on that front page.
Okay, all right.
Yeah, and then the rest were all dilution problems.
Okay, all right. So we don't want to take that one again?
Sounds good to me.
Oh, yeah, I would. Okay, so looking at my list of labs, let's just kind of go down the list. So we know we did the the immunoelectrophoresis, we did that this, you know, with the gel, right? So you remember it was this one, the Edvotech handout. Okay, so that's where let's just start there, because that's on the syllabus first, even though we pushed it down later, because we were looking for 2 days we could do this, and we ended up not really needing the 2 days. So now we know. So the immunoelectrophoresis handout, that's key. I'm gonna mark that and make a stack right in front of my computer here. So remember, that was doing the antibody antigen. We loaded the gel. So the principle behind the test was immunodiffusion, where we looked for the line of precipitin to where the antibody and antigens move toward each other if they were the antigen for the antibody. Remember, we started out by putting the antigen in the wells and ran them across. Is that right? And then we put the antibodies in the trough and let them migrate through the gel for immunodiffusion. So that part, that's, you know, there'll be some questions on that. The next one was, did we do CRP, C-reactive protein? I think we did. So Liza's out. And so I
If
can't,
it helps any, I have that on the first exam, we did pipetting, dilution problems, antibody serial dilution, and ELISA.
okay, yeah, but we did do a C-reactive protein, right? I'm saying I can't find my list of that one. Didn't we do a quick CRP with Stan Bio? I thought we did.
Yes, we did.
Okay, who is that? That's Sharuk, thank you. All right, so CRP. And then I've got I'm looking at my list, we went into ASO. And I just saw it. The ASO, this one, RAPET, I think I made a handout for you. It's Stan Bio 2. This is where we detect the anti-streptolysin O, correct? And then, yes, you find it?
Mhm.
Okay, good. And so I hope that some of you aren't sitting right now and going, ooh, I should have kept a lab notebook, because hopefully you got all this together. Then we went into this one. This one was one of our rushed labs. It was our ANA, right? Anti-nuclear antibody, right? We had some slides. The good thing is we, I don't know if I'm going to use your slides, but I'll use some pictures for you to detect the different patterns. That's kind of our main goal. Along with the reason we do it for systemic lupus, erythematosus, and other autoimmune. So it's not just for that. So you should have a handout right here. So the ANA is definitely on there. And then I had the
But I remember our lab, like it was the first lab, and our slides were very bad.
oh, well, I know they were, but in
Yeah.
this handout, you do have pictures of patterns. And so I'll look for like the obvious, biggest, nicest, real pictures of the really good ANA patterns for you to identify. Because those do show up on the board, and I think I've mentioned that before, and they usually use the fluorescent technique. So they're actually green patterns on the board. But we didn't see that, so we won't use the dye patterns. The next one I had was mono, the heterophil antibody detection mono. Remember we did that one. And so that's infectious mono. And then I don't remember doing I don't think we did the slide X. I think we had that on the list, but we switched out from that. We just did the group A strep test. Okay. Remember that? We did strep test, strep A. So you should have that one. So we'll change that to strep A. And then we did the RPR. We had a one-page RPR, plasma reagent with syphilis, what it's used for, how we use it. Remember, this is a non-treponemal, meaning it's not an antibody produced against the treponemal. It's one of those, it's not the heterophil, but it's another one that's produced, you know, in response, but not actually against the treponemal. So we definitely had the RPR. And then we went into some other tests we brought out. There's my rheumatoid factor, RF, rheumatoid factor. Some of our autoimmunes here. The H. pylori. H. pylori. Remember, only one worked out of the whole group, but we did do the technique and the principle behind it. H. pylori antibody detection. Or was it Lyme disease that didn't work? Which one didn't work? Or both of them didn't work? Remember Lyme disease? From our rickettsial infection, Lyme disease, Borrelia burgdorferi. And lastly was flu, right? Flu A, flu B. So a lot of those tests it's in the principle of the test was the same, but it was just a different serology detection of antibodies or antigens against or those being detected.
Are these on Blackboard to print out? I have all but the H. pylori.
I don't know. I don't think so on Blackboard.
There's only a couple on here. The RPR, the Rimmel color slide for mono, and then the RF Direct are the only ones on Blackboard.
Yeah, the Blackboard. I don't think I gave you even access to a lot of that, but I had a lot of the old labs. I might have access to them for you. Like we didn't do, you know, we just, let's see, got flu. But I don't even know, I could try to get some of these on there, but I didn't. I was just giving them out since we were live. Lyme.
I can look in my other folders. I might have just misplaced it, but I have everything but I guess the H. pylori one. Yeah,
basically what we did was we were going through the old refrigerator, and if we found the kit, we were just printing out the procedure manuals from the kit themselves. I wasn't uploading them into Blackboard. Okay, I think that's the group of stuff. So I've got, starting with immunoelectrophoresis, C-reactive protein, did we do the AS? ANA. Did we do ASO? Was that on here? Yeah, ASO was there. ASO, ANA, mono, strep A, RPR, rheumatoid factor, flu, Lyme, and H. pylori. So that's about what? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 things.
So let's say we're trying to make a Quizlet out of this. Would we just go like put the procedure on there and the principle, or like by.
I wouldn't worry so much about the principle. I mean, the procedure. I would worry more about the intended use, and then mainly what the actual kit was looking for. Like it was like I'm looking at strep A, and I go, it is a direct group A streptococcus antigen test. I would be able to answer that the strep A test is a test looking for the strep antigen. And keep that. That's probably the main thing I'll be looking at when I'm putting those questions together.
And did you say there would also be like pictures?
Yeah, for the ANA, I'm more concerned with you detecting, seeing, but it could be just a picture of, is this a positive or negative test based on some of these.
So will we possibly have like a quiz or a PowerPoint to look at for that?
No,
Oof.
no, just review the labs. There's no quiz. Think of it as a practical, but on Blackboard. So, we're only going to test you over what you did in lab. Not going to pull stuff, out of a test bank somewhere. So it's going to be just here's a immunoelectrophoresis. What are we looking for here? If you had a patient's serum, what are you trying to detect? And how is this interpreted? So that's mainly what I'm wanting you to know. So when I say a strep test, that you should always know that swab is looking for the strep antigen from the throat. If I'm looking at a mono with a blood drop, that with the mono spot, I'm not spot, but the mono, I'm looking for the heterophil antibody against infectious Epstein-Barr virus. With ANA, I need to know the patterns. I need to know what the patterns may indicate. That the anti-nuclear antibody pattern is, if it's detected, what does it mean to have a speckled pattern? And can I recognize a speckled pattern? And so review, so I'm not, you have the handout there has slides. You have the slides patterns, okay? And I'm sure you can Google and get some better ones. But those may find themselves right back on the Blackboard exam. The raw PET, what does raw PET stand for? Don't remember that. I think I pointed that out one day. We finally figured out what raw PET means, because that's what StanBio's does with that's their testing techniques. Anybody remember what raw PET means? And then a little bit about streptolysin O from that. And then the immunoelectrophoresis, it goes into, I think, after you get past like the setup and the immunoelectrophoresis, there's a page written about it. But I'm not going to expect you to, you need to know what we did and how we did it and why we, did all that, and then expected results. So for that one, I would focus on the immunoelectrophoresis background information, and then expected results. And then there's some, for those that are looking for questions, there's some questions on the back. Study questions and answers. Why is the blue dye added to the protein solution for electrophoresis? Why do precipitates form arcs? How does immunodiffusion assay differ from immunoelectrophoresis assay? How many arcs are observed from whole serum samples? What does each arc represent? And what result can you expect if the gel were stained with a protein stain? And all those answers are right there. So you've got, 5 questions with answers on the EdvoTech handout.
Just one more question.
Sure.
Is
I
there
think that covers it
multiple
all.
choice?
What's that?
Multiple choice.
Yeah.
Okay, cool.
How many
Oh,
questions?
that's your
do you
question?
have in
Multiple choice? Yes, it's multiple choice.
Yeah, definitely. That was like a big question for me.
Okay.
I was gonna ask the same question as Allison's.
What's our question?
About, like, do you have in mind how many questions you're gonna have on there? Or
Oh, there is, what did I say, 10? I would expect 25.
okay.
I have
Okay.
11. Is there?
You want 11 questions?
No, I have 11 different things.
Oh,
So
11 different things. What did I leave out?
I don't know. Or did I add something?
I might have not written down. Let's see, I got rheumatoid factor, flu, Lyme, H. pylori, that's 4. RPR, 5, strep A, 6, mono, 7, ANA, 8, ASO, 9, CRP, 10. Did I leave one out? Oh, the immunoelectrophoresis, that's 11. Yes, I left that out.
Okay.
You're right. Sorry, it is 11. So yeah, about 25.
Yeah, I left out ASO.
Huh?
I left out ASO.
I think we did that one. I think we did.
Yeah, we did. I just didn't write it down in my list.
So real basic. I mean, you know, it's just a evaluation of what you got out of doing all these immuno labs, serology labs. Tried to introduce as many as we had and some that we just discovered. So, you know, we look at our schedule. We were hoping we may come across some COVID tests. We didn't. We were hoping to introduce the real-time PCR technology. We didn't get the chance to do that. And then the only other test we had that we could do was pregnancy test. And you already knew the principles behind all that, just detect HCG. Because we go back to, you know, day one where I said, I do never want you to be in the lab and not understand what you're doing. You know, you can just sit and do, but if you don't understand what you're doing, then you're not you know, then you're not valuing your position and your profession. What you're saying is anybody could do what I do. And that's why we use our knowledge. So we want to be able to say, you know, you need me here doing this in case it doesn't work as easy as wave testing, you know, where something is wrong. Maybe I do get the wrong kind of swab. I get the wrong blood. I put whole blood where I'm supposed to put serum, or, you know my results aren't supposed to cause any harm, but I told somebody they were pregnant when they weren't. So we want to value what we do, even if it is doing the wave testing out of a box. But you know, this past weekend I did coagulase test, and you know, yeah, anybody could probably take that out of the fridge and heat it, you know, warm it up and put it on their Staph aureus and see that it coagulates, just to make sure that it is Staph aureus, and it was. So some fun stuff.
I found what RAPET mean.
Yes,
It's Rapid Particle Enhanced Technology.
yes. Thank you. That is their technology that they use to detect things.
It's on the rheumatoid factor handout.
I knew it was on one of them, but
Yeah.
it was labeled on several of them that we use from StanBio.
Yeah.
Rapid particle enhancement technique. Is that it? Say it one more time.
Rapid Particle Enhanced Technology.
Enhanced technology. Okay. All right. Any other questions? Oops, didn't want to do that. Take my participant line down. No other questions. So remember the,
relevant to class.
what's
How
that?
long are you gonna, how long are you gonna be in your office today? This is unrelevant to class.
Oh, I'm here. Yeah, I'm here all day.
My mom said she may have some more test kits for you.
Oh, great. Yes. Perfect. Yeah, we got a donation last week of 5 strep kits. I think it was strep. So we're gonna get to replace the 1998 or the whatever the ones I've been finding. Yeah, we got a fresh batch for next year. So yeah, we take anything, and we'll try to use it, and if we can't, we'll get rid of it. So yeah, we are all about donations. We did get in our I would say our cute little box of parasites. They're here from Puerto Rico. They showed up on Friday, and yes, we have a box of 40 new parasitic samples for you guys to get through next semester.
That makes me so jealous.
Well, they don't look much different than the ones y'all dove in every day. I'm just hoping they're a little more populated than those were. They're supposed to be, so it won't be near the trouble of finding eggs. So they look, they're like this. They're like little bitty. It's a box this big, like 40 samples in one little box. But they are labeled, so I don't have to have a key to know what's in each one. So it's definitely will be a change. All right, any other questions? So remember, both of these are open on Wednesday. So Wednesday, that morning, both these will open. I had to sneeze. Both of these will open early Wednesday and then stay open till 11:59. So you'll have all day to take both. We will not, no class, give you, all day long to get it done. And then on tomorrow for micro, we'll do the same thing. We're going to lecture at 11 and review out what we're going to have on the lab exam on Thursday. And most of that one is going to be the slides, the mycology slides. And
Most or all of it?
most. There are a couple of things that we still need that we can pop on there. It won't be all slides, no. There'll be a few other questions to go along with that group, because that one's going to take me more time to make up. That's why I told Allison I'm going to be in my office, because I've got to get that into Blackboard some way. So that's going to be the toughest part. That one's not there. So, and none of the lab exams are in Blackboard. So these two is where I'm going to spend most of my time before Wednesday and Thursday.
What about the question pools? Do we have an estimate?
Somebody asked earlier. I didn't write it down. It's around 100 for both.
Okay, thank you.
I did a 50 question quiz, and I did reduce the pools down. The pools were way more than that, so I know it's close to 100. I couldn't cut it in half because there were so many chapters, but I cut it I cut it down to be a randomized out of that pool, but I didn't count them, no. And again, there's no matching. No, I don't think there were any true false. I know there was some matching in the pools, but pools, that'll be eliminated come test time. There won't be any matching. Any other questions? All right. Since this is last immuno lecture and somebody wants their picture taken already, I can tell.
Heather, why did your child just lick you?
Don't judge me. I don't know. She's a dog half of the time. Yeah. She wanted to see herself in the camera, so I turned it on, and then of course she would do something like that.
All right, y'all ready? Let's turn on cameras one last time for immuno. And if we don't have you, if we're not following you on Instagram and you want us to follow you, follow us. And if I see you're following us, then I'll get everybody on there best I can. All right, there he comes. All right, love the hat. All right, 1, 2, 3. Big smiles. I don't know who's calling me, but somebody's calling me right now. That's great. Hold on. Just keep them up. Anyway, I have no idea who this is. Somebody calling me. It's a Jonesboro number, 275-4823. Is that anybody's number? All right, they hung up. All right, one more. Let's go. 1, 2, 3. Yay. All right. Hey, it's been a great semester. Keep up. Get this final done. We have final exam, not this week, but next week. It's going to be comprehensive over every exam pool. So nothing new, but it will be pooled from everything you've had all semester. It is comprehensive. So we will see you tomorrow in micro. If not, we'll see you again Wednesday. Well, I won't see you Wednesday or Thursday. So good luck on the test. If you have any questions, I'm here. Text me, Facebook me, email me. Maybe a little slower than that, but if you need me, let me know. All right. So we'll stop the recording.